We are going to do the exercise of emasculation, bagging and tagging. For emasculation, with the help of forceps or scissors, we are going to remove the anthers before anthesis. That means before the pollen grain formation has taken place in them. So we are going to gently remove all the anthers from the flower. Now we are going to cover it with the help of paper bag. This paper bag is made up of butter paper and it should have minute pores so that the plant can respire, the flower can respire. Now we are going to secure it with the help of a paper clip, and then we are going to put a tag on it, and this tag contains all the information regarding this emasculated flower. It has the name of the experiment, date of the emasculation, date of pollination, and name of the breeder. Now we are going to dust the desired pollen grains over it. So we are going to remove the butter paper bag, and then we'll dust the pollen grains over it this way, and then we are going to again cover it with the help of paper bag. And then we are going to secure it with the help of paper clips. And on the tag we are going to write the information, that is the date of dusting of pollen grains. Now controlled pollination, it includes emasculation, tagging and bagging. So objective to perform controlled pollination, that is artificial hybridization through emasculation, tagging and bagging. Requirements: scissor, forceps, brush, alcohol or methyl spirit for sterilizing our hands and other objects. Magnifying glass, paper bags, paper clips, tags, field notebook, desired plant and flowering. Theory: artificial hybridization or controlled pollination is a technique under conventional plant breeding programs for plant or crop improvement in which male and female parent having desired traits are crossed with each other. So a male parent is chosen as well as a female parent is chosen, both of them having the desired characteristic or the superior characteristics. In case the plant is bisexual, then the anthers are removed to make this a female flower. Now anthers, after removal of the anther, which is called as emasculation, now the desired pollen grains are dusted over it. Then we are going to cross both these plants, and then as a result we are going to get the seeds, and from this seed a new plant will be grown, and this plant is going to have the superior characteristics of both the plants. Plant breeding is a science of changing and improving the genotype of plants and production of new varieties which is better than original type. This can be done by emasculation of flowers of a bisexual flower in which the stamens are removed before anthesis to obtain female parent, and pollen from the desired male parent is transferred onto its stigma. Now what do? First of all, we do the emasculation, in which with the help of scissor or forceps we remove the anthers. Then next step is bagging, in which we cover the emasculated flower with the help of a paper bag. Then the next step is cross pollination of emasculated flower, in which we dust the pollen grains of the desired male flower on our emasculated flower. Then we are again going to cover it with the help of a paper bag. This step is called as rebagging, and we are going to put a tag over it containing all the information. This step is called as tagging. Okay, on the tag we put all the information regarding our breeding experiment. Now let's see the procedure. First step is selection of parents. Parental plants with desirable characters or traits must be selected from the local areas. The male and the female plant from the local areas and are supposed to be best suited to existing conditions. Sterilize forceps, scissor, brush as well as hands with the help of alcohol or methylated spirit. Selection of flowers. For controlled pollination, the flower stigma should be receptive and the anthers should not be dehisced. Select a flower in bud condition where anthesis has not occurred. Emasculation. Open the bud carefully and remove the stamens before maturation of stamens with the help of forceps or scissor. Mark this as the female parent. Observe the stigma of the emasculated flower with the help of magnifying glass and make it sure that it does not carry even a single grain of foreign pollen. Next step is bagging. Cover the emasculated flower with a plastic bag to protect it from undesired pollen. The bag should be held securely in place with a paper clip or string or thread. Select the size of a bag in accordance to the size of the flower. Bag must be transparent, made up of butter paper or polythene with minute pores on it. Dusting of pollen grains. Bring into physical contact anthers of the desired male plant containing mature pollen grains with the stigmatic surface of the emasculated female flower. Use forceps or brush for dusting. Rebagging. Cover the pollinated flower again with the bag immediately to prevent contamination. Now the fruits are allowed to develop. Tagging. For identification, label female parent with the tag below flower which contains the information. Number referring to the field record. So on the tag, we put the number of the experiment that we are carrying out. Then date of emasculation, date of crossing or pollination, the name of the female parent, then put a cross sign and then write the name of the male parent. First the female parent, then the male parent, and then finally the name of the breeder. Pods of desirable plants are harvested and after complete drying they are threshed. In the coming season, stored seeds are sown separately to raise the F1 generation. The plants of F1 generation are called the hybrids. So this way you are going to get an image or a setup for your spotting. So you have to write the comments and also draw the diagram and label it. You can label the scissor or the forceps, you can label the anthers being removed and the flower. So the comments you're going to write, the given image shows the setup of emasculation. It is a setup of artificial hybridization. Emasculation is the process of removal of anthers before maturation of stamens. It is done to prevent self-pollination. It is done with the help of forceps or scissor, and in the unisexual flower, emasculation is not required because it has either the male organs or the female organs. Next, this way, this is the image of bagging. So you're going to write the given image shows the setup of bagging. It is a step in artificial hybridization technique. It is done on female flower. It is done after emasculation to avoid cross-pollination by undesired pollen grains. Rebagging is done after dusting desired pollen grains on the receptive stigma of female flower. This is the diagram of tagging. You are going to write the comments this way. The given image shows the setup of tagging. It is a step in artificial hybridization. It is done after emasculation to write details about artificial hybridization experiment. It contains the following information: number referring to the field record, date of emasculation, date of crossing, name of the female plant, then cross sign, and then the male parent and name of the breeder. This way you are going to do the spotting.